Okay, my name is Amanda Garcia. I'm in Dr. Segovia's genetics class. This is video problem number 3. I am doing the application question and problem question number 29 from section 14.2. The question says, how would the deletion of the following sequences or features most likely affect a eukaryotic pre-mRNA? A, an AAU-AAA consensus sequence, B, a 5' cap, and C, a poly A tail. Shown in this diagram is a visual of where exactly all three of these are located and the process of a DNA turning into a mature mRNA. Here you can see the pre-mRNA and the location of the consensus sequence, the cleavage site, and the poly A tail for future reference when describing the effects of these being deleted. For part A of the question, processing of the 3' end of the pre-mRNA requires sequences located upstream and downstream of the cleavage site. Collectively, these are termed polyadenylation signals. The AAU-AAA sequence is also called the poly A consensus sequence, and it's usually 11 to 30 nucleotides upstream of the cleavage site. It determines the point at which cleavage will take place. If this were to be deleted, as asked in the question, in a eukaryotic pre-mRNA, it would prevent the binding of the cleavage and polyadenylation factor. In this case, no cleavage or polyadenylation of the pre-mRNA would occur. For part B of the question, asking about a deletion of the 5' cap, a background of the 5' cap is that it is a cap consisting of a modified guanine nucleotide and several methyl groups and is added to the 5' end. The 5' cap facilitates the binding of a ribosome, increases the stability of the mRNA, and may affect the removal of introns. If this were to be deleted from a pre-mRNA, it would prevent the splicing of the intron nearest to the 5' cap. Also, the stability of the pre-mRNA and its ability to be translated would be affected. For part C of the question, asking about the deletion of a poly A tail, for a background of the poly A tail, it confers stability on the mRNA. It plays a role in its export to the cytoplasm and facilitates attachment of the ribosome. A number of proteins take part in finding the cleavage site and removing the 3' end. After cleavage is completed, adenine nucleotides are added without a template to the new 3' end. This creates the poly A tail. Without the addition of a poly A tail, as asked in the question, in a pre-mRNA, the mRNA may be degraded by nucleases in the cytoplasm since it would not have stability.